Good afternoon. It is my enormous pleasure to be part of this meeting and to try and enthuse about Alan Turing in a rather different direction. And so what I would like you to do is to hold in your mind the fact that this is the same individual we are talking about in all these presentations. So it is the same man. And there's a human side, there's the computational side, there's the foundations of mathematical logic side, and then there is this stuff as well. So this excursion into mathematical biology, and that is the terrific thing about having this day and really what we're celebrating. I'm going to take you on slightly a roller coaster ride. There will be various other personalities who come in and out through the talk. They will not get explained. Their biography will not be given. And they are a sort of part of the story, but essentially this is another group of individuals that Alan Turing is corresponding with and talking to on this sort of very applied mathematical work that also prefigures a lot of what people who work in mathematical modeling or in applied differential equations or applied dynamical systems do, from day to day, and amazingly it pays the bills for us. So here are some pictures that will get explained on the way through. It's essentially a talk in two parts. The first part, there will be loads of acronyms. Okay, I will not apologize for using acronyms because he uses very long words like morphogenesis time and again. So the paper that you may well have heard of, the 1952 paper, that will be the subject of the first half of the talk. And then the second half will be the various other bits and pieces that appear in the collected works. So volume 4 of the collected works, edited by Peter Saunders, looks like this. But its most important and interesting dimension is actually this one. So people on Zoom, it's that one. Okay, it's very thin as a volume. And that's because there is essentially only one published paper, but there is a lot else and archive material that I find terrifically interesting because it's not the finished Proti version. It's not the version where you sort of cover over your tracks and you say, well, of course, it had to be this way all along. So let's make these definitions and then everything unfolds in a really nice way. It's the crossings out, the going back and starting a calculation again, the not getting it right first time, the typos, it's all there. And so there's some extra mathematical interest because this stuff is mathematically much more interesting than the stuff everybody knows. Okay, so that's also interesting. There are loads of figures. These are all taken from the archive material in King's, which is available for you all to look at. So there's a homework element here. Okay, you can go online. They've redone the website. It has a new URL. This is an animation on the new website. It was down for a few months, and that's because they were transferring it over to a new server and so on. So it's all available, and you can browse it. It has some sort of historical signposting and an index as well. So it's a terrific resource, and you can go and read all this material without needing to go anywhere near Cambridge. So I thought, in case I don't make the story particularly clear, I give you the summary. So the summary is on 2 slides. I've hinted at this already. So the chemical basis of morphogenesis is the easy version. It's written for a broad, biologically informed audience, and it's recognized as important and novel as a contribution, but it's hard work for the biologists. And to an extent, it's quite easily criticized. And because of the tradition that it comes from, and maybe the people he was talking to, in some sense, it gets overtaken by events in 1953 and this sense of a revolution in biology. So there's a lot of context. There's a lot of context. So I'm not going to give you all the context, but I may hint at some of it, that there are competing views of, if you like, the philosophy of biology, and the one sort of supported by some of the people Turing talks to is not the one that, in a sense, wins out in the second half of the 20th century. Although, as Stephen has already said, in terms of systems biology, something of that does indeed come back. And so there is a sense of synthesis in the last couple of decades. This is much more interesting. And so he writes down an equation usually ascribed to these people called Swift and Hohenberg in 1977. He writes that down in 1953 or so. And there's a lot of thinking in the notes about things like numerical computation, so solution of differential equations computationally, the role of symmetry in organizing some characteristics of these problems. And these are essential these days, but to have them there in 1952 to 54 is fascinating, really is. So then the more detailed version of this, because of various topics that he's come into contact with previously, describing things in terms of a Fourier space representation of solutions comes very naturally. So Stigler's law is the law that things are named after the last person to discover them, not the first person. So it's another example of that. Obviously, Stigler's law is an example of itself, which is rather nice. So symmetry organizes some of these calculations. And there's an interplay between theory and computation that we completely take for granted now. You read the Philip Bond report on mathematics, all mathematicians must learn to code. Okay, it's absolutely taken for granted that this is our education, and this is where it really starts. There's plenty that is not in here, so one needs to be careful not to ascribe to Turing ideas that he did not invent, as it were. And I wrote a paper outlining something of this in a few years ago. So that will come back, and that will be the end of this presentation as well. So to try and give you a timeline, I'm going to sort of do something slightly forensic, and really this is, as a talk, it's all the production of some detective work that I've been sort of enthused and delighted to be able to undertake in various archives and reading various documents, including stuff that you can go and check to see that I've got it right or disagree with me about. So there's a letter preserved in the King's Archive to John Zachary Young. He is the University of Bath connection in all this. So I'm going to make a little plug for Bath. We gave him an honorary degree. But the fact that this happened approximately 15 years before our institution was founded means I had to work a bit to get a University of Bath connection into this talk, but there it is. So he says, at the moment, I'm not working on the problem of the relation between the logical and physical structure of the brain. So there are questions about how many neurons does the brain contain. That feeds into Stephen's articulation of the number of bits one might need in order to produce sufficiently complicated storage or processes to generate something that would be machine intelligence. But he has a mathematical theory of embryology, which looks like it will solve this whole host of problems. So gastrulation we'll say something about later. Leaf arrangement is the classic one, the Fibonacci series, as Debbie has already showed us up there on the banknote. Color patterns and patterns on nearly spherical structures, such as some radiolaria, and I'll say a little bit about that. This is more difficult and doubtful, hence I will give it to my MSc students to work on, because it's the most difficult bit. And he's doing this because he's getting somewhere with it. Managed to get a new computer. Slightly later, I think, than the 12th of February, but there we are. And then the first hint of what he's up to, we have in an exchange of letters between Alan Turing and Philip Hall at King's. He's elected a FRS in March '51. Very gratifying to be about to join the Olympics. I hope I'm not described as distinguished for work on unsoluble problems, which is rather nice. So we have a letter that contains some mathematical detail. And really, this is filling in some of this 2 years, 2 months gaps here in the timeline. And Andrew has already given us a sense of the man in that time. So this is the original letter. These references are all to the King's Archive material to help you find this more easily for yourself. I will read this in case you're not as au fait with Turing's handwriting as you would like to be. Waves on leopards are rather more elementary. The leopard skin, before the spots arrive, is suppose an infinite thin sheet containing two chemical substances with concentrations U and V which react and diffuse. This gives you equations of the form du dt is f of u and v plus mu del squared u, dv dt is g of u and v plus mu del squared v. It's the London Math Society, so there will be equations. I should have said that at the start. You may approve, you may disapprove, but there will be equations. And they will tend to look like this. If you take f and g to be linear, near u0, v0, and the whole system to be just unstable, you find, if the constant satisfies certain inequalities, that u equals u0 plus alpha z, v equals v0 plus beta z, where z satisfies a wave equation. Del squared z is 4 pi squared over lambda squared z. The wavelength lambda, depending on the reaction rate fg and diffusion rates mu. What particular solution you get will depend on random disturbances just before instability started. Roughly speaking, you get a random solution. By assuming there is black when z greater than z0, yellow for z less than z0, you get very reasonable leopard skins. Certain slight variations of assumptions give you giraffes, zebras, cows. Cows are dappled. So hence the pictures in the report. So however you look at it, this is a paper that a lot of people cite. Okay, this is the citation plot from 1953 up to this year. So this is 2 months worth of citations of this paper, the chemical basis of morphogenesis. So it clearly has some kind of tangible sort of hold on the mind of people in the applied maths, mathematical biology modeling community. So there are various excerpts of it here. And it's morphogen is a word that he introduces in this paper. So a system of chemical substances called morphogens reacting together and diffusing through a tissue is adequate to account for morphogenesis, the emergence of biological form. So, clearly a huge challenge. It's a modeling paper, so I'll have a mathematical model of a growing embryo. This model will be a simplification and an idealization and consequently a falsification. Okay, so these are words that are music to kind of applied mathematicians' ears. This is what we spend our time doing, and wonder why anybody pays us to produce falsifications the whole time. It is to be hoped that this is kind of good enough. Okay, and then there are various things here. It's a paper in a biological journal, so he starts by reminding readers what complex numbers are. Okay, it's not written for mathematicians. And so he then reminds you of various properties of sums of powers of roots of unity, and he's going to use these later in the paper. There's an interesting paragraph here where he sort of sets out your kind of roadmap through the paper depending on how much mathematical training you have. So if you struggled so far with the complex numbers, then you should do 3, 4, 11, 12, and so on. If you just about understood the complex numbers and the sums of the roots of unity, then do 7, 8, and 9, and the others, they're the maths bit. Okay, so here's your route through it. The central message is just as outlined in the letter to Philip Hall. So 2 chemical concentrations, reactions, spatial diffusions, small departures from equilibrium grow over time and give rise to these kind of patterns with a wavelength to them out of a uniform initial state. This is interesting because when you read it carefully, you say section 6 isn't mentioned. So that's interesting, even though the paper clearly has a section 6. And section 14 is mentioned, but it does not exist in the paper. So already there's some, something to get your teeth into here about what happened. Okay, and then there are some lovely plots. So this is what he means by a stationary wave. In the simple case, we've got sort of, 2.5 wavelengths here that have kind of emerged out of a nearly uniform chemical concentrations with a kind of spatial grid here, and then the heights of these columns are the concentration level. These were obtained with the computer. He did some of them by hand, and then there's some hypothetical chemical reactions to try and support how this might actually look in terms of real chemicals. You could, without needing obviously to trouble any health and safety considerations, mix up at home and try and get something like this. And then there's a long page here about where he's going in the future, including nonlinear equations, and that's probably the poorest slide in the entire talk in terms of award of text. So I did the obvious thing, which was to go to the Royal Society and say, tell me about the paper that you received. And they do keep lovely records. So here it is, page 77 of this register of papers. And it's paper number 62, submitted in 1951. It's been entered in the logbook. And some referees were chosen. So A.B. Hill is the hill associated with Hill functions. Sort of rational functions of concentrations that describe inhibitory or autocatalytic processes in chemistry, and Lancelot Hogben, who wrote a textbook, popular science book, Mathematics for the Million, that sold extremely well. They were chosen as referees, and unfortunately, they both declined to provide a report on the paper. So they sent the paper back, and they found 2 other referees. So the grandson of Charles Darwin and JBS Haldane, who essentially knew about everything. One of his students described him as the last man to know everything. That's obviously a field-dependent quote. But these two actually deigned to provide reports, and the Royal Society kept their reports. So we have that annotation here. This is scanning further along the page from the piece I showed you before. So we have dates and we have names, and we have this sort of little annotation, no rep here. So they really didn't provide a report, although they were available. And that is some initial indication of quite how far from, if you like, mainstream thinking the paper and the work it described was. So this was not a sort of a butterfly collecting, count the numbers of specimens type paper. This was something really different. What did the referees make of it? Referees' comments always worth reading. So Darwin says this paper is well worth printing. The danger is that it will be taken too literally. I love this. The mathematics are not very deep. The mathematician will not get any new principles from it. So that's rather lovely. And then there's some interesting discussion of what Turing meant by the term bilateral symmetry that appeared in the version sent in. And I think that's where the editing issue probably came from, actually. So the sections that were missing, the numbering that didn't quite follow, I think that's connected with this. Slightly a red herring. And then the whole day, this is, classic referee material. You've either read or written precisely this in your time, I would say. I regard the central idea as being sufficiently important to warrant publication. I'm equally clear that the paper should not be published as it stands. Okay. And then there's 4 pages, and he objected enormously to that sort of level of biological detail, the examples that Turing gave, because he said, if you're writing for a biological audience, they will know all this. So that was interesting. And this is part of that kind of interdisciplinary, tussle that we always have. And then there's also a comment about this guy, Pontecorvo in Glasgow. And that maybe explains why Turing in his Manchester office had a copy of a paper by a biologist from Glasgow. There's no obvious other connection, but Haldane, I think, puts them in touch. And indeed, they correspond. Haldane appears to have lent him a book. And the revised version essentially goes through Haldane for publication. So what happened? Okay, the paper is accepted for publication. It comes out, and copies, reprints are circulated in the spring of '52 to a number of people, including Pringle, who's a zoologist in Cambridge. And it clearly excites some interest in the community. So it really is something that people are talking about at the time. And so this is the opening paper in a symposium volume published by the Society for Experimental Biology, who hold a symposium event in Oxford in July '52. And there's a sort of summary of some of the contents of the paper. And the context is interesting because in that paper, Pringle starts off by reminding people of Lotka-Volterra type dynamics, populations, and from which he says some interesting results emerge. And that idea of competitive interactions is clearly in the air as well. And then he goes on to some detailed discussion of the 1952 paper. But by detail, he only gets as far as the first part of it. Okay, so he doesn't get into the diffusion part. It's only the reaction terms and the linearized stuff here that gets summarized. So there's already a sense that people are picking this up and saying it's interesting and it's novel, but I don't quite understand the message. But he does say this discovery opens up a new field of speculation and experiment. So there's clearly plenty more to do here. There's a letter that Kings have from Conrad Howard Waddington, who's another leading zoologist, particularly interested in fruit flies, Drosophila. And he says it is very encouraging in his letter that some really competent mathematician has at last taken up this subject. And then we get the admission, although parts of your discussion were rather above my head, I found the general arguments extremely interesting and suggestive. He's a little bit worried about various things. The fact that noise triggers these instabilities seems to imply that the details of how they form is a little bit random, and that's not good for developmental biology because you need some predictability to always build the same structure. And then he sort of says, go and read the kind of stuff that I read, looking at butterfly wings. There's a series of articles by Henke with lots of plates with pictures of different patterns on butterfly wings. So he suggests to go and for Turing to go and read those. He slightly hardens his stance later. So this is a book from 56. And there he sort of says this does not seem very convincing, and he gives some biological insight into why that would be. So Hydra is a sort of small worm-like structure, and flatworms, similarly, if you kind of cut them into bits and you ask them to regenerate, then they have some better idea of the position of different organs and the structure of their body due to the boundaries and where they expect the boundaries to be, rather than through some intrinsic chemical wavelength. And so Turing's idea is that there is some intrinsic wavelength given by the reaction rates and the chemistry that you set up, but there's something missing in terms of development. So here, Waddington's comment, rather the pattern must arise as a whole within the boundaries of the material available. So there's clearly more to do. And so Waddington is nervous about, in a sense, ascribing too much to Turing's mechanism, interesting and suggestive though it is. There's a colleague of Turing's at Manchester, Wardlaw, who is very enthusiastic. It's almost as if he's been looking for this as an idea. And up to the point that Turing's paper is published, he's sort of slightly bundling around in the dark, and then he says, one day light began to break through, and he has a number of conversations with Turing. And in almost every essay he writes afterwards, he cites the 1952 paper. Okay, this is clearly a huge supporter of this, maybe particularly because Turing frames some of the later work in terms of phylotaxis, so the Fibonacci series organizing patterns of seeds in a sunflower head. And so it's a more botanical example compared to the zoological examples that we've had previously. But this is a collection of people who don't particularly need to talk to each other. Okay, if you're working on fruit flies, you don't have much to say to people working on potato tubers at this stage. Somehow there's a paper here that is trying to talk to both groups, and that's really challenging. So a kind of living geometry, and he thought through that there must be some common underlying principles. So he's really taken with this to the point that they must discuss it in the lab. Okay, so this is a group of people working on things like potatoes, and they cite the chemical basis of morphogenesis in their theses at this point. So they've clearly read it, and so I have this sense that it has some circulation within that that part of the biology department in Manchester. And he says it's not inconsistent. And this sort of goes to a wider context. So this sense, going back to, well, shortly after the origin of species, for example, of what it means to actually be alive. Is there a sense of a spark of life that makes living matter different in nature to kind of dead matter? Okay, and this is, I'm going to characterize this in 5 seconds as mechanism versus vitalism. Okay, and there's the beginnings of a third way in the development in the philosophy of biology that actually there could be something slightly more than just mechanism, because organisms come in as a whole and they have some internal organization. And so that sense of the structure needs to be consistent and coherent across the whole animal body. And that's a more subtle idea. Various people have written on this. Evelyn Fox Keller's book is the one I would recommend, which is why I've highlighted it slightly here, in terms of understanding the sweep of 20th century biology. But Wardlaw cannot resist many times, and often in the original German, quoting the plant forms cells, not cells the plant, which is an interesting way of thinking about that relation between individual cells and the body that they sit in. So what's the motivation then for the chemical basis of morphogenesis? He's not the first person to write down the sort of reaction diffusion stuff. Okay, so this is a chap called Reshevsky working in Chicago who writes a lot about sort of, what's he call it, physicochemical biology or something. It's a collection of kind of ideas in biophysics where he's trying to work out how to apply physical principles to understand the structure of biological organisms. And this is better and a huge improvement on Darcy Thompson's On Growth and Form, which is essentially about measuring stuff. Okay, this is much more about the internal structure and organization and dynamics and therefore growth of things. So he's not the first person to write down this, but there is an enormous need for mathematicians to get involved. And this is recognized, for example, by Waddington, the fruit fly guy, before the Second World War, who writes things like this. So organizers and genes is a sort of a key item in this sort of sense of developing developmental biology. And he tries to sort of formalize things like gastrulation, so the way that material moves around, with little arrow diagrams like this. Okay, he sort of hopes that this could be maths, but he's not really sure. And it's not maths. Okay, it's clearly not maths because he doesn't do anything. He doesn't tell us, you know, this is an operator, that this is really just layers of cells moving around and folding. And he kind of knows it's not maths. And so there's this great quote. This example is not, of course, offered as a serious attempt to formulate an adequate or appropriate set of symbols. It will have fulfilled its function if its inelegance annoys some mathematician sufficiently to provoke him to head and so on. Okay, so I think this is lovely because this is the gauntlet being thrown down. Okay, and gastrulation is, you know, according to some people, the most important time of your life. Okay, so a little definition here. This is going from a bunch of cells that have just proliferated in the very early embryo, and they start forming into distinct layers, and then you get orientation in that bunch of cells, and that allows you to actually start development and stem cell differentiation because you know where your head is and where your tail is, and where your front is and where your back is as an organism. Okay, so gastrulation is the most important time of your life. There's another interesting sort of subtle idea around in the landscape, which is this idea of inhibitory influences, and this is really a cartoon diagram that this guy, Vincent Wigglesworth, he only has one figure like this. It appears in many papers, so I've cited it from 1940 onwards. You know, figures are hard to make, okay? So, and the idea is that this is a two-dimensional surface, and there are little kind of local concentrations here that somehow sweep in, advect chemical concentration from the nearby surroundings. And so you get an inhibitory effect because you can't form another little local high concentration very close to the next one. And he says, this is rather like universities and students. Okay, I thought this would appeal to this audience. So if these students are all you know, drawn off, sucked in, signed up by an existing university, then that will inhibit the appearance of another university in the neighborhood of that. So you end up with some kind of spacing selection principle for these peaks. And then he says, of course, it's equally possible that a university produces some offensive substance that pushes other universities out to a certain distance. So I've quoted this one, which is actually a little later. This is the 1959. He develops this analogy delightfully from one paper to the next. It's the same idea just presented, and I couldn't resist putting that in. So there's a sense here of a spatially inhibitory mechanism that might also give rise to some characteristic spacing. And this is 1940. This is well before chemical bases of morphogenesis. So I have then a hypothesis, which essentially is that Turing read a copy of this book. Okay, so this is this symposium from the Society of Experimental Biology, Volume 2, Growth. So this is the papers read in Oxford in July 47 that include these. The Richards paper, we know that he read it because not only did he cite it, but he wanted to reproduce a figure from it in one of the unpublished figures. And then there's a paper by the Snows on phytotaxis. There's a paper by Spiegelman, which is very interesting as well, thinking about actually organization within a cell, but it's a similar collection of ideas around autocatalytic chemistry, inhibition, competition, these Lotka-Volterra equations. And there are papers by a whole host of people who I've mentioned so far, and we're in Turing's orbit, if you like. So there's one volume here that contains all the ideas that go into the chemical basis of morphogenesis, apart from writing it down in sort of decent, understandable mathematical notation. So as a motivation for the chemical basis of morphogenesis, I think this is a runner. a contender, if you like, in terms of where do these ideas come from and the context of the conversation that was circulating at the time. So I'm just throwing that out there. I'm going to skip over this because time is unfortunately against me. And I should say something about the later program as well. So going back to the Red Book has the paper we've just discussed, and then in 3 parts, there's morphogen theory of phyllotaxis. So clearly something to discuss with Wardlaw and to make sure is botanically grounded. It comes starting with the geometry, the organization of these buds around the stem. It then moves into the chemistry, and then finally into the mathematical modeling. And it's really on the mathematical modeling that I'm going to focus. But I'm going to give you a thumbnail sketch of the previous stuff. Again, it's in the King's archive, and it's those pages, and at the start of this outline of development of the Daisy manuscript that I feel should be extended further. And so it's some of those calculations that we might get to, depending on how the time goes. And so then there's 4 equations to kind of remind ourselves that this is the London Mathematical Society, not the London Botanical Society. So how do you understand Fibonacci numbers? Well, you take a plant stem, you unroll it, and you imagine that the left and the right end would be glued together around the stem. This is in the collected works. And then you look at the lattice of these points, imagining that then could theoretically tile the plane in a periodic fashion. And that periodicity corresponds to the wrapping it up around the stem. And as that plant grows, you then shift through a collection of periodic arrangements of these points. And that shifting through moves you through the Fibonacci series as your sunflower accretes more seed heads. So this is a kind of dynamic version of how these lattices evolve. There's some commentary by Jonathan Swinton on this. And because it's in the first part of this 3-part article, I will refer to as MTP. It's reasonably well elaborated by Turing himself. The second part goes back to chemistry and says, what are the most general chemical reactions that I could get that would try and do this, this sort of generation of these periodic structures, building on the chemical basis of morphogenesis paper. So he says they must look like this. This is some kind of diffusion between cells. So we've got morphogens labeled by the first index and cells labeled by the second index. So I have some discrete reaction diffusion set up here. And then there's a homogeneous equilibrium. I've got to think about small perturbations to that equilibrium. And the only wavelengths which are of interest are either very long waves, so very small wave numbers, or ones near to some optimum value, 2 pi over k0. So I'm now heading mathwards, okay, unapologetically. So these concentrations, essentially, I'm going to write them in terms of some eigenfunctions taken care of by the W's, and then some amplitudes, V and U, that correspond to those different kind of modes of operation. So either I'm thinking about modes of response that have a positive wave number, or the 0 wave number mode. So the one with the maximum is being at a positive wave number is U, and the long wavelength mode is V. And quadratic interactions should be enough, and indeed are the only ones that are chemically feasible. So I'm only going to include quadratic nonlinearities here. And then these things he writes down later in terms of spatial gradients. So he then has some further arguments to knock out a bunch of those terms. So these are the equations I showed you before, but with some space. And then sort of essentially, this is the V thing is diffusing rapidly. And so we will assume that this is locally always in equilibrium. So there's actually no time derivative here. And then it's just the V equation that is dynamically evolving, again, to try and simplify the problem. The third part, the part that he gives to the MSc students, Bernard Richards, is concerned with doing exactly the same thing, but on the surface of a sphere. So in your eigenfunction expansion, you write everything in terms of spherical harmonics. And that enables you to understand some of these icosahedral, dodecahedral, symmetric objects, which are very tiny marine organisms that build a calcium shell. And I think this is the calcium shell, roughly speaking. There are enormous numbers of these pictures collected as people did in the, well, what in the UK is the Victorian era, as part of conquering the world, really. So back to the two variables. This is a dispersion curve. This is wave number on the horizontal axis, range of shortest lattice vector, against growth rate. Okay, so there's a local maximum of growth rate at 0 wave number, so this long wavelength mode, and then there's a maximum here at a positive wave number corresponding to the pattern forming instability, we would now call it. So he combines these and then starts to write down an explicit form. And so what he's done here is to take something more complicated, much more complicated than in the CBM paper, and essentially wrapped up all the CBM behavior into this first U equation. So what am I going to do now? I'm going to skip onto this, because this is the point at which you have something here that is generating a linear term with spatial derivatives that generates that pattern forming instability at a positive wave number. And then you have something really complicated going on here because of that UV interaction that he's decided is always at equilibrium. And so you have to invert the spatial operator there. So there's actually a non-local term that he's bolted onto the end here. So this is a terrible fourth order non-linear PDE for U of X and T with a non-local term. Okay. So this is actually much more complicated than the thing that these guys, Swift and Herbo, wrote down in 1977. He then does a classic applied maths thing, and he non-dimensionalizes, which is just lovely to see, because it's the kind of step that everybody goes through. You rarely bother talking about much, but he doesn't. And then he says, okay, what I want to find actually are solutions that are periodic on, in a sense, the simplest lattice possible, which would be a hexagonal lattice in the plane. It also seems to be the one that enables us to understand these changes when you dynamically evolve the lattice and you move between lattices that have different basis vectors, but you always move in such a way as to generate the next number in the Fibonacci sequence in real space. So there's a kind of selection principle there, which I'm not going to talk about, but the hexagonal lattice is the one that seems to be the kind of the decision point where you move from one lattice to the next. So the hexagonal one is interesting. And then he does this in sort of delightful detail. So it's 7 Fourier modes. You have a uniform field here, psi, and then you have some etas, which have wave numbers that are spaced equally at 60 degrees around the unit circle. So you plug that in, and then you do a kind of vicious Fourier truncation. Okay, so you look at the quadratic terms, and you throw out everything that then has a wave vector that is not either 0 or something that fits on the unit circle. And then he looks at stability. And so this is exactly what we would do. Unfortunately, as a technique for this problem, because he's picked something that is terrifically complicated, it falls down. And it falls down actually in a moderately obvious way. This is a sketch of what he's trying to get to. Okay, so this would be a hexagonal plan form. And you see this when you look at the reduced equations for those modes. So if he's taken the 6 modes around the unit circle, be equal magnitude now. So we've got the 0 mode psi, and we've got the pattern forming mode eta, and they interact like this. So V is psi squared plus 6 eta squared. H is a parameter, I is a parameter, it appears in both equations, that's fine. But actually, the parameter sigma that describe the strength of the or the range of the non-local interaction, that's gone. Okay, so unfortunately, as a Fourier truncation, this doesn't capture all the complexity of the model that he started with. But it does. I mean, there's a sense here of that joke where you're looking for your keys and you look under the light, and somebody asks you why you're looking under the light for your keys, because they know that you dropped them over here. And you say, well, I'm looking under the light because I can see here and I can check to see if my keys are here. Because he's doing this because he can. But unfortunately, it's now lost some mathematical connection with the problem that he set up and the problem he wants to compete with. But you can do this. And so what's interesting is that we can still do this and understand it. You can solve it terrifically. So in deriving some of these expressions, there is a page of algebra that is nowhere to be seen, but he's written down the answer. And when you go through the calculations, you do indeed check that that is correct. And what he comes up with is encapsulated in the IH plane here with this mysterious figure called figure X. Okay, it doesn't get much more mysterious than that. So what is this? So there's a curve here. Okay, and that curve is the envelope of a collection of straight lines. Let's do it to the way you're looking at it, which corresponds to constant ratios of psi and eta. So you pick a ratio of psi and eta, that gives you the existence of solutions on one straight line in the IH plane, and then you sort of vary that ratio and you get a whole family of straight lines. That family of straight lines has an envelope to it, and what he's deduced here is the envelope. So you've got a collection of solutions over here to the right of that curve, and they are generated by the formulas that we saw before. So you can do this in a kind of modern way. So I'm fixing an H and plotting I against solution amplitude here. And so this is what we call a bifurcation diagram. So you have a collection of equilibria. The ones that are of interest are the solid lines. And you can recover figure X by plotting the location of these turning points on the left as the solutions come back. So in modern terminology, that would be a saddle node bifurcation, and this will be a curve on which those saddle node bifurcations lie. So we can understand the analysis, but unfortunately, because of the Fourier truncation problem, this has now become disconnected with the model that he started with. So this is again a restatement of the equation. If you wrote that in a slightly more proper form, so the 1 minus sigma squared del squared inverse is acting on the u squared, you would say that I've got a convolution type term here, and that convolution is well known. And before I read this stuff, I realized I'd actually written a paper on the one-dimensional version of this. So I thought I'd put the reference in. So this is now what we tend to call the Swift-Sternberg equation, this first piece, with a non-local term that is motivated by any number of phenomena that essentially have a fast-slow timescale structure. And so something relaxing to equilibrium fast that you then adiabatically eliminate in favor of a non-local term. So this is a well-studied thing. It all leads on to a theory of bifurcation, which is a word that Turing doesn't have, and how those calculations are organized by symmetry. And they're organized by symmetry whenever you have a kind of symmetric pattern that you're starting with. I appreciate time is moving on. I have to show you this because it's a delightful comment by Ian Stewart, latterly of Warwick, about the role of symmetry in dynamics. So symmetries complicate things and simplify them. They complicate them by introducing exceptional types of behavior, increasing the number of variables involved, and making vanish things that usually do not vanish. They simplify them by introducing exceptional types of behavior, increasing the number of variables involved, and making vanish things that usually do not vanish. So you have to change your bifurcation theory point of view to look at patterns, and we now understand something of the standard bifurcation structure for problems that form hexagonal patterns. Hexagons are one part of the classification, if you like, and here is a kind of typical bifurcation diagram of the kind that Turing was exploring. And we can do more modern, slightly more precise things to simplify the equations. Believe it or not, these three equations are simpler than the two at the top, and we call that progress. And I had an undergraduate student working with me a few years ago looking at that. And we also understand a great deal about the formation of these hexagonal patterns in general. So there's a lot of kind of modern dynamical systems theory that comes into play to understand, for example, how you can form little patches of hexagons that sit on a flat background. So you don't have to tile the plane with your hexagons. Sometimes you can hold them in tension with a kind of uniform state outside. And there's plenty of interest there. Some of the other material in the archive is his numerical method, which is what we now call a spectral method. You expand everything in Fourier series. He does this fully, as it were. These are sums over many of these amplitudes, and has some notation here for how the nonlinear terms are going to go. He makes some approximation of a polynomial in terms of something trigonometric to simplify that when he comes to implement this. And then the implementation he does as we would exactly for a spectral method. Time stepping is slightly more complicated, and I'm not sure it's actually time stepping, even in an Euler sense, because the time step depends on the wave number that you pick. So there's something slightly more subtle going on, but you could certainly see this as trying to iterate to find an equilibrium solution. So as long as this iteration converges, you would be able to pick up equilibria by doing this. It's a very numerically challenging PDE, particularly for the resources he has available. And so I'm amazed that he gets something out that has some kind of hexagonal symmetry to it, but clearly also feels the effects of this non-locality, and so it's modulated over longer length scales. So this is precisely the kind of picture that you would expect to see given our understanding of the problem that he's doing. So here's the summary again, and I apologize enormously for going over time. Chemical basis of morphogenesis is easy. It's written for a broad biological audience. It's important and novel, but it's overtaken by things like, the double helix and the excitement around actually understanding the structure of DNA, the fact that that gives rise to an easy replication mechanism, and the excitement around molecular biology. And that happens shortly after this is published. The later work is mathematically much more challenging. It prefigures a lot of things from the 70s, 80s, 90s that have kept people like me paid for doing these modeling exercises that are easily falsifiable. And it also hints at some of the theoretical issues that underpin some of these and are also trends in modern applied mathematics. The archive material, I think we can extend this thing called outline of development of the DAISY as it appears in the Red Book a little bit further. And that's interesting because that actually captures more of the mathematical argument. So the mathematics is more complicated, and also the organization there makes that clear. So there are various things that were worked on. There are some interesting parallel developments in the fluid dynamics literature that I haven't talked about at all, but some of the same ideas get explored at very similar stages in the context of things like Rayleigh-Bernard convection. There doesn't appear to be much crossover between those two literatures at this point. Obviously, things kind of come together in the early 70s in a much more organized way. And I've written about this, including, maybe more interestingly, the appendix is my reconstruction, going further of this outline of development of the DAISY, complete with typos, both mine and his, to sort of try and show how this could have gone, what this would have looked like. Thank you all very much indeed.